Good day, everyone. I'm Kathleen Joyce, Austria, a fourth year biology student, major in zoology, under the guidance of Assistant Professor Sara Maika Dalisay and Assistant Professor John Michael Castro. This is my thesis proposal entitled Yolk Absorption Dynamics and Utilization in Leopothera plumbeus as a basis for its larval rearing protocols. So, some of you might have seen this fish before. This is a union, a native freshwater fish in the Philippines known for its delicate, flavorful flesh, typically enjoyed as dishes like pinaksio, kinamatisan, or even dai. A union is an endemic and commercially important freshwater fish in the Philippines, but it faces a serious conservation crisis because it is now classified as vulnerable, wild catch statistics showing a severe 68% decline from 2002 to 2024. This highlights the urgent need for effective captive breeding strategies. The major challenge in a union aquaculture is the high larval mortality during the critical period. This is the phase where the larva successfully switch from relying on its internal yolk sac to consuming external food. And when yolk sac depletes before external feeding, larvae reaches the point of no return or the stage when they experience irreversible starvation, thereby leading to death. A 2020 study by Garcia and others identified an extremely brief critical period that is a 12-hour window from 40 hours post hatching to 52 hours post hatching. This is defined by two key points: yolk sac exhaustion at 40 hph and the PNR with oil globule exhaustion at 52 hph. They also found out that larvae fed at 46 to 48 hph had significantly higher feeding incidence, thereby inferring that the time to first feeding must be within this 2-hour interval window. This table shows us the developmental stages of a union larvae from 0 hours post hatching to 72 hours post hatching, or the pre-flexion larval stage, having a mature mouth gape. Now, I want to emphasize that mouth gape size is a crucial factor in their high mortality rate. This is because the union larvae are size selective feeders. At 48 hours post hatching, its mouth gape is estimated to be between 0.12 to 0.17 millimeters only. That is in proportion with the total length of 2.83 millimeters as shown in this figure. Hence, only selected microorganisms like the Haline rotifer, Brachionus rotundiformis, and Artemia or brine shrimp nopli are some of the ideal feeds for a union larvae at 1 to 13 days post-hatching and 14 to 35 days post-hatching, respectively. Now, although previous studies have determined the ideal time for first feeding and the rate and efficiency of yolk resorption, there is a lack of mathematical modeling for the correlation between yolk resorption, feeding success, and growth potential. They have established the survival deadline or PNR at 50 hph, but not the optimal feeding time to optimize larval growth. Hence, lack of comparative data or temporal feeding onset within the viable window. To put it simply, we know when the larvae must eat to survive, but we do not know the optimal time to feed them to maximize larval growth. This is the focus of my study. That is to relate the yolk absorption kinetics of a union larvae to the optimal timing of first exogenous feeding by achieving these specific objectives. First, through yolk kinetics, we aim to determine the utilization pattern of the total endogenous reserves that includes both the yolk sac and oil globule. And then we will be computing for the following parameters. Next is by determining the time to first feeding, or TFF, assessing the feeding incidence and gut content analysis. Lastly is by evaluating the effects of different feeding regimes on survival, specific growth rate, and the starvation sensitivity index or R ratio. The study establishes scientific data-driven Ayunian hatchery protocols. Defining the critical feeding window increases survival, optimizing growth, and thereby providing precise live feeding timing guidelines for hatchery managers. This supports sustainable captive breeding conservation and thereby the economic viability of this native species. Here's an overview of my methodology. First would be the broodstock collection and maintenance of mature union. This shall be sourced from before and there will be a period for acclimatization and a separation of sexes, such that during fertilization there will be a 1:2 female to male ratio. Females would then be injected with the HCG hormone to induce spawning, followed by egg incubation and placement of newly hatched larvae in 10-liter basins with a stocking density of 5 larvae per liter. Now, to properly monitor the yolk kinetics and feeding incidence of a union, 6 feeding regimes will be employed with 3 replicas. 10 samples will then be collected per 2-hour intervals from 40 to 50 hours post hatching. This will be preserved and mounted on glass slides for microscopic imaging. 6 key parameters will then be observed and measured, such as yolk sac volume, total length and weight, growth rate, survival rate, and feeding incidence. Finally, data will be analyzed using one-way ANOVA and Tukey's post hoc test to determine significant differences across 6 treatments. Mature broodstock of a union will be sourced from BIFAR, the Provincial Fisheries Office of Laguna, located in Sampaloc Lake in San Pablo, Laguna. This study will employ a complete randomized design involving 6 treatments with 3 replicates, testing 6 feeding times starting at 40 hours post-hatching to 50 hours post-hatching, or the previously established critical period, with 2 hours intervals in between. And then, since a union larvae were found to have the highest feeding incidence at around 46 to 48 hours post hatching, treatment 5 will be the positive control or the standard feeding protocol. And shown underneath are the water volume, which is 10 liters, and the stocking density with 50 larvae per basin, all in all, that is approximately 5 larvae per liter, and the controlled conditions, dissolved oxygen being lower than 5 milligrams per liter, and temperature being 27 to 29 degrees Celsius, and by having consistent light exposure. So as for the feeds, once larvae reach the 40 hours post-hatching stage or the start of yolk sac exhaustion, this will be fed with green water or chlorella, followed by copepods and Artemia nopli, which are just enough to fit in their mouth gape. And then destructive sampling will be done, wherein 10 larvae will be collected per basin, and the following parameters will be measured. So as mentioned, yolk sac volume, oil globule volume, total length, and weight through water displacement. These measurements will then be calculated to obtain the weight-specific growth rate, the yolk utilization efficiency, the survival rate, the feeding incidence, and the starvation sensitivity index, or R ratio, which in this formula shows that R is equal to the T resorption, or the time from hatching to yolk exhaustion, over the time from hatching to first successful feeding, or the T onset. So, our expected output is a clear definition of the critical feeding window that maximizes growth efficiency in a union larvae. By using one-way ANOVA and Tukey's post hoc tests to compare the mean weight or the means of the weight-specific growth rate across the treatment groups, we will be able to determine the growth optimizing feeding time to be supported by the other parameters we measured. So here is my project timeline from May to July, which may be subdivided into 4 phases: the preliminary phase, the setup and hatching, intensive experiment, and post-experiment. Here is a closer look for the preliminary phase. Basically, this will involve the securing of permits and other procurement of necessary materials, followed by setup and hatchery, intensive experiment, and the post-experiment analysis, which will all be done during my mid-year from June, ideally from June to July. Here is the proposed budget for this study. And here are my references. That's it. Thank you for listening.